Hello guys, welcome back to my lesson. So today we're going to do questions under the topic of DNA code of life. So let's look at the first set of questions. The diagram below represents two stages of protein synthesis. Those stages are transcription and translation. If you look at that diagram, the whole box is a cell, and in that circle is the nucleus. The question says provide labels for bond at X. Bond at X is the bond between amino acids, that is peptide bond. Organelle 4 is the nucleus. Molecule 1, that is a DNA molecule because it has two strands. Molecule 5 is tRNA or the transfer RNA. Let's look at the next question. It says that does strand 2 or 3 serve as a template for the process that is taking place? If you look at the line that is between the two strands there, the horizontal lines are facing, those small horizontal lines are facing strand 2. It means that they're pairing with nitrogenous bases of the second strand. So this means that strand 2 is the answer. So the next question says write down the DNA based triplets that correspond to the codon numbered 7. The codon numbered 7 is GUA. So you're going to use the sequence that I always use. You write DNA first, under DNA you write mRNA, under mRNA you write tRNA. So you're going to substitute GUA at mRNA and then you find your DNA. The answer is going to be CAT, I mean. So let's look at the next question. It says that the table below shows two differences between DNA molecule and an RNA molecule. We have the type of sugar, we have the number of strands. So if you fill this table, let's look at the question. Write down the letters A to D in your answer book and list the differences. So sugar at DNA is deoxyribose sugar, at RNA B is ribose, at C how many strands? In DNA we have 2, in RNA we have only 1 strand. So let's look at the next set of questions, and the diagram below shows a process occurring in the nucleus of a cell. So if you look at this diagram, we can see a DNA molecule unzipping, and then it says that identify the process shown in the diagram. You can only see a DNA molecule, so a process whereby there is only a DNA molecule, that is DNA replication, because that's where it makes a copy of itself. So this process is DNA replication. The first question says identify the process shown in the diagram, that is DNA replication. Name the bonds that are labeled at X. You know, in DNA molecule we have a weak hydrogen bond. Name the phase in the cell cycle in which this process takes place. Interphase. State 2 reasons why the process mentioned is important. It doubles the genetic material, it keeps chromosome number constant, it creates genetically identical daughter cells. So let's look at the last set of questions we have. It says that the diagram below represents a DNA replication. So if you look at the first DNA molecule, which is X, it has the nucleotides there pairing. The second one, we have strand 1 and 2. The third one, 3 and 4. If you look at the nitrogenous bases at 3 and 4, we have G pairing with G there in the middle, and which is an error. And then they say that name the phase of the cell during which DNA replication takes place. That is interphase. Identify the type of the error occurred in strand 3. Instead of C, there is G there. That is gene mutation. Give base pair showing error referred to in question 2.2. You can see that G is pairing with G, so that's what you're going to write. The next question says describe the consequences of the error during DNA replication. You're going to write that an incorrect nitrogenous base sequence results in a different DNA strand, and then this will result in a genetic code for daughter cells formed, wrong genetic code, and then different proteins could be formed. So now let's look at the next question. Says tabulate differences between DNA replication and transcription. So this question is repeating. Remember in the last video that I made, we found this question. So in DNA replication, adenine bonds with thymine. In transcription, adenine bonds with uracil. In DNA replication, an identical DNA molecule is formed. In transcription, an mRNA molecule is formed, right? In DNA replication, both strands act as templates. In transcription, one strand of DNA acts as a template. So that is the last question. Thank you so much for listening. Don't forget to subscribe to the channel. I would really appreciate that. Thank you.